Good morning, students. I asked you to look up some things. Tell me how many of you found the time to actually look up all those questions that I was mentioning yesterday. Raise your hands, please. Just the two of you. Satyakam and Gopika, nobody else. Vishantan, theek hai. Ashi, okay, good. Nobody else. We are approximately 12 or 13 of us. अच्छा ठीक है, so we'll go one by one, but this time I want you to turn on your camera, please. We'll start with Satyakam. Satyakam, बताओ.
Hello, ma'am.
Hi.
Ma'am, that this you asked what does that indicate that mitochondria and chloroplast contain their own DNA and 70S ribosome. So, ma'am, this shows that sometime in the uh, like earlier they used to be independently living like organisms, and later they evolved into a cell, most probably.
Why would this have happened?
Ma'am, because of we have studied that things evolve, so maybe because of.
Benefit क्या होगा Satyakam?
Ma'am, because of more complex life, like for example plants and animals, but they can't just have mitochondria and 70S ribosomes. They need a much more organelles like we have right now.
अच्छा, अब Satyakam, please turn on your camera.
Oh, ma'am, I can't turn on my camera because there is some error with my Teams. I don't know, ma'am. Earlier, I can't even open the microphone. Then I reinstalled it.
अब तुम्हारा ये issue ठीक होगा. चलो ठीक है. Now Gopika, tell me what did you find out? हाँ.
Yes, ma'am.
Gopika,
So for
camera
the
please.
first, um,
Yes, tell me now.
so for the first one, it was the for you asked about compartmentalization. So the answer is that no, they do have compartmentalization, not as organized as you, but it exists. For the second, there is that they do have cytoskeleton elements, which are exactly similar functioning ones to use, like tubulin and fibrin, I think. And for the third one, it indicates a symbiosis at some stage in evolution.
Okay. ठीक है. Good. Thank you. Dhwani.
Yes, ma'am. So
Dhwani, I
about
would
the
like to see you as
mitochondria
well.
and am I visible?
Yes, you are. Tell me, Dhwani.
Yesterday I read about this term, the endosymbiont theory, which basically said that initially mitochondria and chloroplasts were individual bacteria by themselves, and a eukaryotic cell engulfed them, and a relationship began such that uh, since mitochondria were so efficient in in consuming energy, therefore the cell had a lot of use
Consuming
for that,
energy.
and
Dhwani,
uh, I mean, in
yeah,
terms of
generating.
like generating energy. Sorry, yeah. So that, and because of that, this relationship began, and thereafter we've finally evolved with mitochondria as proper organelles. So therefore, now you won't see mitochondria or chloroplasts as individual free living bacteria, because they've also developed a dependence for us, and therefore this is known as the endosymbiont theory.
Good. Yeah, good. Did you also see something about compartmentalization or any other aspects? So I wanted to know from you if the concept of pro and eukaryote has changed slightly after the class, at least. when during the school times you hear that there is no cytoskeletal element and there is no compartmentalization, there are a lot of recent discoveries which actually refute some points. Okay. Anybody else? और किसी ने हाथ उठाया था? विशंतन ने? किसने? गोपिका, you can lower your hands. गोपिका, कुछ और कहना है क्या?
नहीं,
नहीं.
sorry.
हाँ विशंतन, बोलो.
Ma'am, am I audible?
You're audible, but Vishantan, I would like to see you.
Ma'am, I am using a PC right now because my laptop isn't working, so it doesn't have a webcam.
ठीक है, बोलो.
Ma'am, so basically, like regarding the cellular compartmentalization, so like recently, not that recently, but like comparatively recently, like scientists discovered things like carboxysomes and like micro compartments in bacteria, which are like basically protein shells which enclose specific enzymes and like other proteins, and like really small and primitive compared to the modern cell organelles that exist in eukaryotes. And basically like they serve like pretty primitive functions. Like carboxysome is used for I guess carbon dioxide fixation, or maybe carbon fixation in general. And like you have certain gas vesicles which are sort of like vacuoles, but they contain specific gases which provide buoyancy to aqueous bacteria.
Yes, so these were the inclusion bodies that we mentioned, right? Carboxysomes and gas vesicles, there were magnetosomes and so many more storage structures as well. But you still don't call them as organelles. You just call them as inclusion bodies or vesicles or just structures. Okay.
Yeah.
ठीक है. Anything about the endosymbiotic theory?
Ma'am, it's pretty much like as Dhwani and Gopika
What
mentioned,
Dhwani
that
said.
is,
Okay.
yeah, that's
हाँ.
what I read.
Okay, so the similarities that I mentioned between the mitochondrial features, the chloroplast features of the chloroplast, and the mostly the gram negatives because of the presence of porins and the other features, they indicate to some evolutionary event wherein there must have been some endosymbiosis with one organism being engulfed by the other organism. And this was majorly to delegate functions. So mitochondria would do just so this particular organism which then turned into or transformed into a mitochondria would produce energy. Chloroplast would fix carbon dioxide for food. So this was called as endosymbiotic theory. But I'm not sure if anybody of you saw that this is quite a debatable theory. I mean, it's not all for, everybody is not all for this theory. Did anybody go through the thought process which refutes this theory as well? Nobody?
Ma'am, I saw an article
हाँ.
in which one person quoted that what if
पहले बोल कौन रहा है? एक मिनट. Who is it?
ma'am Satyakam.
Satyakam. हाँ, tell me Satyakam.
So ma'am, one person quoted that what if there is some this like fusion of DNA in mitochondria which we are not sure of, and maybe cause some crippleness in the body.
सत्यकम, मुझे समझ में नहीं आया. फ्यूजन ऑफ बैक्टीरिया, फ्यूजन ऑफ डीएनए मतलब
I mean,
क्या रेलेवेंस? हाँ.
basically mutation in some of those genes.
ऑफ माइटोकॉन्ड्रिया.
Yeah.
ओके. माइटोकॉन्ड्रियल डीएनए के बहुत सारे एप्लीकेशन है. मतलब इवन फिंगरप्रिंटिंग में और आइडेंटिफिकेशन में कई सारे वो हैं, एडवांटेजेस हैं इसके. Anyway, so maybe in the next class, all those who did study the endosymbiotic theory can also go through what are the limitations of the theory. Why is it that it's not an acceptable theory as of until date? We don't say that it's an acceptable theory. With regards to the first two questions of whether or not there is compartmentalization and क्या था दूसरा question? What was it?
Cytoskeletal structures.
करेक्ट. साइटोस्केलेटल एलिमेंट्स. तो साइटोस्केलेटल एलिमेंट्स तो मैंने मेंशन कर ही दिया था कि एनालॉगस हैं बहुत सारे. or we are still looking for more? I mean, we will be talking about E. coli's division, and there we will see how FTSZ and REB, they help in exactly the same manner like tubulin and actin would do in a eukaryotic cell. But I have to mention that these most critical features which differentiate the pro and eukaryotes, for example, the nucleus, the a true nucleus, let's say, there are organisms, the prokaryotic organisms, which have a rudimentary kind of nuclear membrane or some kind of an envelope surrounding their genetic material. There are exceptions that have been observed. Then there are also exceptions wherein you find a linear chromosome, which are actually characteristic of eukaryotes, but you find them even in some prokaryotic cell. Again, you will see that many for what were the other feature, like circular DNA, linear DNA ho gaya. Then one, so you talk of haploidy in prokaryotes, and there are organisms reported, prokaryotic organisms reported, which have more than one chromosome as well. So what I mean to say is the boundaries are quite blurred. You cannot say that the prokaryote is a very simple organism with absolutely no differentiation and stuff, and it's only the plasma membrane doing the functions. There are reports, and it is coming up. I mean, we are trying to find out more in depth, but the boundaries are really blurred now. So having said that, I told you that I will be talking a little about the plasma membrane. Let me just first share it. Okay. So the cellular membrane, it refers to the membrane that surrounds the cell as well as the internal membranous system found in cells. Now, whether it is the envelope of organelles or like you saw in the chloroplast, you had the thylakoid membrane system. All these are a unit membrane. Now, I know that this topic will be a part of your biochemistry course, but I'm taking it up for the simple reason that cell membranes, they play a crucial role in the success of prokaryotes in various environments, especially in the extreme conditions. So I wanted to just talk about how organisms adapt the plasma membrane based on the conditions. And for this, I need to really talk about the structure of the plasma membrane. And also, like I said, archaea have major differences in terms of the plasma membrane when you compare archaeal plasma membrane to the bacterial plasma membrane. Now, just by its location, you can understand that it serves a variety of functions like the selectively permeable barrier. And then it is also involved in compartmentalization, so separate organelles dedicated for a particular function. Now I'm going to talk about plasma membrane in general, pro and eukaryotes, and then I'll specifically talk about the prokaryotic cell membrane. So compartmentalization actually separates organelles dedicated for a particular function, and their content is separated from the cytosolic environment. That's the whole idea here. So an organelle could contain specific enzymes which are for a particular purpose, and hence called, for example, mitochondria would have ATP synthase, chloroplast would have rubiscose, so on and so forth. Now, as the plasma membrane or the cell membrane is the outermost covering for many types of cells, it also perceives signal from the environment and then transmits it inside the cell. Besides, it's also the site of action of various reactions, including synthesis of ATP, like you saw in the mitochondrial inner membrane. There were ATP synthases which perform the function of generation of energy. ATP क्या है? Energy currency of cell, ये पता होगा, है ना? Now, let's come to some of the general features of the plasma membrane or the unit membrane. By now, you know that it's a lipid bilayer impregnated with proteins. Now, the major constituents are the phospholipid molecules, which are actually amphipathic. That is, they have a hydrophobic and a hydrophilic part. Now, the subunits, these subunits, whether they are phospholipids or other lipids, they are associated non-covalently. And this has a lot of significance. In fact, it is a prime contributor to its fluidity and some of the other features also that we'll talk about. It is selectively permeable, of course. Then it has asymmetry in terms of the two faces of the membrane. So they're not identical in structure as well as in function, and there is a particular inverted comma में sidedness for both proteins and lipids in the membrane. And finally, the membrane is self-healing or self-repairing structure. So after any kind of damage or a puncture, it rapidly restores. And here again, the non-covalent association helps. Now coming to the fluid mosaic model, which was given by Singer and Nicholson way back in 1972. While there were many models proposed even before this, Singer-Nicholson's model happens to be the most accepted even till date. And this figure that you see is actually taken from the science paper of Singer and Nicholson in 1972, except that this one is colored from this paper. Otherwise, it's exactly the same. The model says that the membranes are made up of amphipathic lipids, which spontaneously associate as such bilayers. And in these bilipid layers, you can see that there are proteins which are associated with the polar head group of lipids, either, so let's say only on the surface, or they span the entire hydrophobic matrix. This kind of a structure was called as fluid mosaic, with fluid being the lipid component and mosaic the protein distributed in this fluid matrix. So I don't know how many of you have seen, I don't know if we still have those, but mosaic is a kind of flooring. So this impregnation of these small chips leads to a mosaic structure. And this is where the proteins contribute to this appearance of mosaic in the plasma membrane. Now, the model proposed that both lipids and proteins are in constant motion with three main motions. Number one, rotational, with both proteins and lipids being able to rotate on their axis perpendicular to the plane of the membrane. Then the second type of motion is the translational diffusion, as it is called, wherein both lipids and proteins, they diffuse along the plane of the membrane. So it's not a static model. And the third type, called as the flip-flop movement or trans-bilayer diffusion, trans-bilayer, across the bilayer, was believed to be theoretically possible, but was not practical for the polar lipids and proteins because of this energy barrier due to the hydrophobic core. Collection of motions is referred to as the fluidity of the membrane. When you say fluid, so you can see here lateral diffusion taking place. Of course, the rotational movement is not clear here. The lipids in the membrane are of three types. There are phospholipids, which are the major component. Then there are glycolipids and also sterols. In fact, the lipids make up about 50% of the total mass of the membrane. Now we'll talk especially about phospholipids. The phospholipid molecules, they have a backbone of glycerol. Then they have a phosphate group and two fatty acid chains. Glycerol, as you can see, is a 3-carbon molecule, and fatty acids are attached to the first and the second carbon. The fatty acid chains can be either saturated or unsaturated, which affects some properties of the membrane, and we'll see what this is in a while. The phosphate group is attached to the third carbon of the glycerol backbone. So here it is glycerol 3-phosphate. And there are variable head groups attached to the phosphate. These two fatty acid chains that you see form the hydrophobic part, represented as these two chains here. And the phospholipids, they are named after their head groups. So there could be, for example, phosphatidylcholine, which has a choline head group here, or phosphatidylserine, or phosphatidylethanolamine, so on and so forth. In the case of glycolipids, you have sugars attached to lipids. And glycolipids make up like 2 to 10% of the membrane mass. Again, here it will be the sugar moieties which form the hydrophilic part. I'm sure you're aware that fatty acids with one chain, they are cone shaped and they spontaneously form such micelles. Now, when they're two fatty acid chains, for example, in phospholipids and in glycolipids, that makes it bulky, and the structure then is of a cylinder. So just by virtue of being cylindrical, both the phospholipids and the glycolipids, they spontaneously form such bilayers in aqueous environments. This energetically favorable structure has the hydrophilic heads facing water on both sides, and the hydrophobic chains are shielded from water. Now, with this kind of a bilayer arrangement, there is a spontaneous closure of the phospholipid layer to form sealed compartments. The closed structure is stable because it avoids the exposure of the hydrophobic hydrocarbon tails to water, which would be energetically unfavorable. The feature of semi-permeability of the membrane is attributed to the structure of phospholipids. Because the interior of the phospholipid bilayer is occupied by hydrophobic fatty acid chains, the membrane is impermeable to water-soluble molecules, including ions and most biological molecules. While lipids are responsible for the fundamental structure of the membrane, it is the proteins which are responsible for carrying out specific membrane functions. Like I mentioned, mostly plasma membranes consist of 50% lipid and 50% protein. Singer-Nicholson model describes two kinds of proteins: the peripheral proteins, which are associated with the lipid components, and the integral proteins, which are anchored in the membrane. Also, the peripheral proteins could be released from the membranes using relatively gentle methods, while the integral proteins, they require a harsher treatment for separation. Many integral proteins are transmembrane proteins which span the entire lipid bilayer with portions exposed on both sides of the membrane. A channel protein, it's an example of an integral protein that selectively allows particular materials like ions or specific kinds of biomolecules to pass into or out of the cell. Now, I mentioned that there are different kinds of motions or movements possible for both lipid and protein moieties, but not all proteins are able to diffuse freely through the membrane. In some cases, what happens is the motility of the membrane proteins is restricted by their association with the cytoskeleton, as you see in the figure here. The Singer and Nicholson model has actually undergone modifications in the last few decades. It has been realized that there's a high density of proteins in the bilipid layer, in contrast to the sparsely present proteins in the earlier model. Then we also know that there are proteins which are only temporarily docked onto membranes. So otherwise they would be in the cytoplasm. The Singer-Nicholson model, you saw the figure earlier, is rather planar structure and does not exhibit large heterogeneities in its surface except these obstructions or these bumps which are caused by the proteins. However, it has been found that the bilayer has heterogeneous patches or domains which are enriched in certain kinds of lipids and proteins. All the different molecules that make up the plasma membrane, they are not evenly distributed across the surface. Instead, there are patches that are more ordered and tightly packed. So, for example, there could be lipid patches which are called as lipid rafts. And finally, there is data to support that the transbilayer or the flip-flop movement of lipids is also possible. It happens in the membrane. So now the model looks somewhat like this. It is not as symmetrical or homogeneous as earlier proposed by Singer and Nicholson. And we know that it's an uneven curved kind of structure with, you see, the dominance of proteins here. The saturated fatty acid chains, they have no double bond. So the chains lie straight, they pack tightly and interact much with each other. However, the fatty acids of most natural phospholipids, they have one or more double bonds. Now, most fatty acids in nature are cis fatty acids. Trans are rare to find. Okay, maybe you know the reason, but I would mention that cis fatty acid is the one which introduces kinks or such bends. The trans would not. Okay, and you can try drawing the figure and understand this. The trans fatty acids are abundant in fast foods, but rarely in nature. Okay, they lie more straight with the saturated fatty acids. अब इससे क्या होता है किंग्स प्रोड्यूस होने से द किंग्स एंड अनसैचुरेटेड सिस फैटी एसिड्स मेक देम लेस टाइटली पैक्ड एंड द ग्रेटर अमाउंट ऑफ स्पेस मेक्स देम more fluid in comparison to a more viscous membrane which is all saturated tails. And this is how the kinks or the bends or unsaturation affects the melting temperature of fatty acid. The introduction of a double bond renders even the long chain acids liquid. Thus, the 18 carbon saturated stearic acid, which has a melting point of 70 degrees centigrade, when you compare it to oleic acid, which contains same 18 carbon atoms, but has one double bond, has a melting point of only 14 degrees centigrade. Okay. Also, the chain length plays a role in fluidity. A shorter chain length reduces the tendency of the hydrocarbon tails to interact with one another, so that the membrane remains fluid at lower temperature. Now, because of its rigid structure, sterols, they play a distinct role in membrane structure. In fact, they have a bidirectional regulation role. Depending on the temperature, the sterol has distinct effects. So, for example, at high temperature, the cholesterol interferes with the movement of the phospholipid fatty acid chains, making the outer part of the membrane less fluid. On the other hand, at lower temperatures, however, sterol has the opposite effect. By interfering with the interactions between fatty acid chains, sterol actually prevents membranes from freezing and maintains membrane fluidity. Under the condition of a change in temperature, the fluidity of the membrane is affected. So at high temperature, you will see that it becomes too fluid, while at lower temperature, it becomes too rigid. Now, unlike eukaryotes, microorganisms or prokaryotes that are exposed to such temperature variation, they adjust the fatty acid composition of their membrane lipids to maintain a relatively constant fluidity. And this they do by making changes in three components. It could be the hydrocarbon length, primarily these three. It could be unsaturation degree, the degree of unsaturation, how many double bonds. Then they also play with the sterol concentration. And in some cases, you will see that the charge and the head group of the phospholipid also has a role to play in maintaining the fluidity. Now, talking of some specific cases here, as the temperature falls, for instance, fatty acid with more cis double bonds are synthesized. So the decrease in bilayer fluidity that would otherwise result from the drop in temperature is avoided. Then shorter and unsaturated fatty acids, they present lower melting points when compared to their respectively longer and saturated counterparts. Adaptation to cold shock has been studied in E. coli and Bacillus subtilis quite extensively, and it has been found that the cold actually induces a phospholipid desaturase, the enzyme which is responsible for introducing unsaturation, the double bonds. While E. coli, it responds to a decreasing growth temperature by increasing the amount of unsaturated fatty acid. What B. subtilis does is it induces the production of these desaturated, the desaturases, which leads to an increase in unsaturated fatty acid. Now coming to the Listeria monocytogenes, I don't know how many of you know, it's a foodborne pathogen. It is also capable of growing at refrigerating temperatures, and it contains an unusual fatty acid profile dominated by more than 95% branched chain fatty acid. This is a kind of adaptation for it. Certain bacterial species belonging to the genera Pseudomonas and Vibrio, they possess an alternative adaptive mechanism when growth is inhibited. They are able to isomerize unsaturated fatty acid from the cis configuration to the trans configuration of the double bond without a shift in its position, just isomerization. So this is with respect to the adaptations that you find in some bacteria. Now coming to some of the archaea, and as I mentioned earlier, most of the extremophiles that we know of, they belong to archaea. So I'm going to point out differences between the plasma membrane of archaea and the bacteria. I'm only going to focus on the plasma membrane and not the other features which also contribute to the survival of archaea in such harsh conditions. There's a sharp contrast between the lipids of plasma membrane of archaea and those of bacteria. So much so that it has been termed as the lipid divide. Now let's start with the first point here. You will notice that here there is an ether linkage instead of an ester linkage that you saw in the case of phospholipids in the earlier phospholipids that I showed you, which had ester linkage between the glycerol and the fatty acid chain. Here there are ether linkages. Now, ether lipids are always present in archaea that reside in high temperature environments without exceptions. But the mesophilic archaea, those that grow and survive in normal conditions, the mesophilic conditions, they also have ether lipids. So this is not directly related to thermophily per se. Now in some archaea, this is that of bacteria and eukaryotes. So you see this lipid bilayer wherein the two fatty acids, they face each other of both the faces. You will notice that in archaea, there is actually a monolayer structure composed of tetraether. So two ether bonds here and two here. Tetraether bipolar phospholipids are found. And here the C40 hydrocarbon chain, so this is basically 20 and 20, C40 hydrocarbon chains, they pass across the membrane bilayer. Thus, tetraether lipids link the two faces of the lipid bilayer covalently, making it all the more rigid, makes the membrane rigid. This structure allows membranes to tolerate extreme conditions. This has been known to contribute to their survival under extreme conditions. Another feature is that the archaeal ether lipids, they are synthesized from, this D-glycerol is incorrect, okay. They are synthesized from glycerol 1-phosphate. Now the enantiomeric difference of the lipid backbone glycerol 1-phosphate and glycerol 3-phosphate, the latter in bacteria, is insignificant in terms of the thermal adaptation again, because enantiomers, they have equivalent thermal properties. But this is an important point wherein here you find glycerol 1-phosphate forming the ether linkages with the fatty acids. Another major difference is associated with the side chains themselves. So you will see that there are unbranched fatty acids in bacteria and eukaryotes, while in archaea there are isoprenoid chains, and they can have these branching side chains as well. So these are the four main aspects, the four main differences that you observe between the plasma membrane of an archaea and that of a bacteria, and some of which, like I mentioned, contributes to their survival in extreme conditions. So with that, I would like to stop the presentation, and I would like you to ask me questions now, because that is all about the plasma membrane that I wanted to discuss. You are going to study, I hope you are going to study the plasma membrane in much more depth in your biochemistry class, but this was just the basic that I wanted to give you. Now I'm open to questions. Any doubts? Please let me know. हाँ विशंतन बोलो.
Ma'am, in the archaeal monolayer, like,
हाँ.
does it induce any other differences apart from like the rigidity? Like the porosity is
Yes, yeah, exactly.
compromised
That
or
is
something?
exactly what I wanted to say. It makes it, of course, it compromises with the porosity, because especially under these extreme conditions, you have to really maintain a balance when things don't just free flow across the membrane. So it does, but I cannot specifically think on the top of my mind about the kind of biomolecules which are affected, but definitely it affects the permeability. ठीक है, anybody else? ये समझ में आया कि ये कैसे अडेप्ट करते हैं ये तीनों फीचर्स से. सो अगर चेन लेंथ तो क्या होगा अगर चेन लेंथ लॉन्ग है तो रिजिड होगा या फ्लूइड होगा? लॉन्गर द चेन लेंथ, मोर रिजिड द मेम्ब्रेन और फ्लूइड द मेम्ब्रेन? बताओ कौन बताने वाला है? कौशिक, कशिश?
More fluid. Am I
Longer
audible?
the chain length, more fluid. Kashish, please explain कैसे?
Am I audible?
You are audible. Tell me, कैसे? Explain your answer. यार ये चला जाता है हमेशा. जस्कीन?
Yes, ma'am.
Jeskeen, longer the chain length, tell me the effect on fluidity.
Ma'am, it will decrease.
अरे डिक्रीज कैसे बोल रहे हो तुम लोग? कशिश, कशिश और जस्कीन मुझे बताओ कि लॉन्गर द चेन लेंथ तो तुम लोग कह रहे हो कि फ्लूइडिटी विल, इट विल बी मोर फ्लूइड.
Ma'am, because a shorter chain length will also increase the fluidity because it will reduce lipid interaction in
हाँ करेक्ट. तो
different
शॉर्टर चेन
layers.
लेंथ विल इंक्रीज फ्लूइडिटी, राइट?
Yes.
बट लॉन्गर चेन लेंथ क्यों बोला कशिश तुमने कि लॉन्गर चेन लेंथ विल इंक्रीज फ्लूइडिटी?
Ma'am, because I thought that if the chain length will be longer, so interaction would increase definitely.
करेक्ट.
But as the length is longer, so it should means like particles have more space to pass through, so it should be more permeable. So the chain should be a bit more fluid, I guess. I thought something like that.
नहीं
Means
नहीं
the
नहीं
particle means the fluids and lipids have more space to flow. I thought in that way.
नहीं 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 बिल्कुल नहीं. हाइड्रोफोबिक बैरियर तो रहेगा ही, चाहे शॉर्ट लेंथ हो चाहे लॉन्ग लेंथ हो, लेकिन जितना लॉन्ग होगा उतना इंटरेक्शन विद द नेबर्स विल बी हायर, राइट? सो इनफैक्ट
Yes.
जब हम ये स्ट्रैटेजी, इसीलिए मैं तुमसे पूछ भी रहे हैं क्योंकि ये एक स्ट्रैटेजी है बैक्टीरिया की या प्रोकैरियोट्स की कंट्रोल करने की. सो द मोमेंट देयर इज a higher temperature, they would start increasing their chain length. And I wanted to know if this makes sense for you now, having told you कि जितने ज्यादा chain length और जितना orderliness होगा, जितने interaction neighbors से होंगे, neighboring fatty acid से होगा, उतनी fluidity decrease करेगी. समझ में आया जस्कीन?
Yes, ma'am.
तो जैसे ही टेम्परेचर इंक्रीज करेगा, अब मुझे तुम लोग बताओ कि इन तीनों पैरामीटर में अगर टेम्परेचर इंक्रीज हो रहा है तो हाउ वुड अ बैक्टीरिया चेंज इन टर्म्स ऑफ दीज थ्री पैरामीटर्स? जस्ट दीज थ्री पैरामीटर्स टेल मी कि टेम्परेचर इंक्रीज हुआ है, हाउ विल अ बैक्टीरिया एडजस्ट? रेज योर हैंड्स प्लीज. और इन्हीं तीन पॉइंट्स के हिसाब से आप मुझे बताओगे कि चेन लेंथ का क्या होगा, अनसैचुरेशन और स्टीरॉल का क्या होगा अगर टेम्परेचर इंक्रीज कर रहा है. टेम्परेचर इंक्रीज कर रहा है मतलब मेम्ब्रेन और फ्लूइड हो रहा है. अब आप मुझे बताओ, who is going to raise hands? Sarthak, do you want to attempt this?
Hey, hello, ma'am.
हाँ सार्थक, tell me.
No, ma'am. Actually, I don't know.
ठीक है, कोई बात नहीं. Others? ऋतुराज?
No, ma'am.
नहीं. और लोग बोलो, रुद्रांश वगैरह बोलो. तो तुम लोग क्या समझे फिर अभी तक? मैंने आप लोग को जो पैरामीटर्स बताए, अभी जब हम चेन लेंथ भी एक्सप्लेन कर रहे हैं तो आरिश बताएगा. आरिश बोलो.
हेलो.
बताओ.
मैम अभी can't
क्लास अटेंड
explain
किया था?
right now.
क्लास अटेंड किया था? अभी जो मैंने बोला वो सब
मैम
सुना
मैंने
था?
लास्ट आज का अटेंड करा था, लास्ट 2 क्लासेस अटेंड नहीं करा था.
नहीं, आज की, सिर्फ आज की बात कर रहे हैं.
हाँ
किया था ना? अब मुझे बताओ
हाँ
कि चेन लेंथ की बात हुई कि अगर चेन लेंथ लॉन्ग होगा तो इंटरेक्शन ज्यादा होगा, करेक्ट?
यस मैम
अगर इंटरेक्शन ज्यादा होगा तो रिजिडिटी इंट्रोड्यूस होगी।
हाँ
ठीक है। अनसैचुरेशन में क्या था? अगर सिस बॉन्ड्स होंगे, सिस डबल बॉन्ड्स होंगे तो क्या होगा? रिजिडिटी या
डबल बॉन्ड
फ्लूइडिटी?
एक मिनट मैम मैम रिजिडिटी होगी
अगर सब उल्टा, क्या कर रहे हो तुम लोग, क्या बोल रहे हो मुझे? अगर सिस बॉन्ड्स हैं, डबल बॉन्ड्स, तो रिजिडिटी इंट्रोड्यूस होगी। हम यहाँ पे आप लोग को किंक्स दिखा रहे हैं, हम आपको यहाँ पे ऑर्डरली स्ट्रक्चर दिखा रहे हैं कि कैसे ये देखो, ये क्या है, क्या हो रहा है एक डबल बॉन्ड से, क्या हो रहा है बताओ? तभी मैंने कहा कि ट्रांस डबल बॉन्ड्स नहीं होते हैं, सिस डबल बॉन्ड्स। अगर तुम ट्रांस का फिगर बना के देखो तो इट विल बी क्वाइट अ लीनियर स्ट्रक्चर, लेकिन हमने यहाँ पे तुम्हें किंक बना के दिखाया कि ये देखो कैसे ऑर्डरलीनेस और ये इफेक्ट होता है। क्या कुछ भी समझ में नहीं आया तुम लोग को? कोई और मुझे बताएगा? प्रथम, बंसल? वेरी गुड, तुरंत मीटिंग लीव कर दो। हाँ गोपिका?
यस मैम।
इस फिगर से क्या समझ में आ रहा है गोपिका? मतलब इसलिए हमने दो दो तीन तीन चीज बना बना के एक ही चीज को दो बार तीन बार समझाया। बोलो सिस से क्या होगा? सिस,
अनसैचुरेशन।
हाँ अनसैचुरेशन।
हाँ सर, देयर डबल बॉन्ड्स तो फ्लूइडिटी आएगी।
करेक्ट, क्यों आएगी?
बिकॉज इन केस अनसैचुरेटेड है, मतलब देयर नो डबल बॉन्ड्स, तो विल बी
क्योंकि
वेरी क्लोजली
ऑर्डरलीनेस
पैक्ड।
इफेक्ट हुआ है।
हाँ, तो ऑर्डरली हो जाएगा एंड उस केस में रिजिड हो जाएगा।
हाँ, अगर वो स्ट्रेट सैचुरेटेड चेन्स हैं तो वो ऑर्डरलीनेस इंट्रोड्यूस कर रहा है, और वो अगर किंक्स हैं तो इट डिस्टर्ब्स दिस ऑर्डरलीनेस। करेक्ट। कौन बचा? भव्या, मुझे बताओ कि टेम्परेचर डिक्रीज करेगा तो स्टीरॉल कॉन्सेंट्रेशन इंक्रीज करेगा या डिक्रीज? भव्या?
टेम्परेचर कम होने पे स्टिरॉल ज़्यादा होने।
और क्यों, ये भी रीजन भी बताओगे? टेम्परेचर कम होने पे मेम्ब्रेन की फ्लूइडिटी कैसे अफेक्ट हो रही है?
टेम्परेचर कम होगा तो बॉन्ड स्ट्रांग होंगे।
टेम्परेचर कम होगा तो बॉन्ड स्ट्रांग होंगे, तो स्टेरॉल्स इंक्रीज करके कैसे फ्लूइडिटी को मेंटेन कर सकते हैं?
इंटरेक्शन ज़्यादा हो गया तो फ्लूइडिटी कम होगा।
हम्म हम्म तो कोलेस्ट्रॉल स्टीरॉल्स जो हैं वो सर्व करेंगे एज अ बैरियर, ओके, टू ब्रेक दीज इंटरेक्शंस एंड मेंटेन द फ्लूइडिटी। ठीक है। एंड जब हम डुअल रोल बता रहे थे तो यही स्टेरॉल्स हाई टेम्परेचर में कैसे फ्लूइडिटी मेंटेन करते हैं?
हाई टेम्परेचर में ये हाई टेम्परेचर में बॉन्ड ब्रेक होंगे
हाँ,
तो वो फ्लूइड ज़्यादा होगा।
तो स्टेरॉल से क्या होगा?
ये डिस्टरबेंस रेजिस्टेंस।
नहीं, स्टेरॉल्स एक्चुअली rigidity देंगे. It's more like adding to this fluid structure. Here they start imparting some kind of rigidity in this very fluid matrix. जो हम बाई डायरेक्शनल रेगुलेटर कह रहे थे वो ये था. ठीक है, म्यूट करो आशी.
यस मैम।
टेम्परेचर लो है, चेन लेंथ कैसे अफेक्ट होगा? अगर टेम्परेचर लो हो गया है तो चेन लेंथ कैसे अफेक्ट होगा?
अगर टेम्परेचर लो होगा तो चेन लेंथ कम होना चाहिए।
अडेप्ट करते वक्त कम होना चाहिए, क्योंकि लॉन्गर होगा तो वो और रिजिडिटी देता जाएगा, है ना, इंट्रैक्शंस बढ़ते
हाँ
जाएंगे. ठीक है. ये कितने लोगों को समझ में नहीं आया? जरा प्लीज मुझे हाथ रेज करो, तो हम जरा हैंड रेज करो, हम देखें, हम समझाने की कोशिश करें. क्या नहीं समझ में आया? ये तो बहुत छोटा सा टॉपिक था. हैव आई आस्क्ड एवरीबॉडी? ध्वनि,
Yes, ma'am.
टेल मी व्हाट इज नॉट क्लियर.
Ma'am, it was clear.
आर दीज थ्री फैक्टर्स क्लियर कि ये कैसे अडेप्टेबिलिटी में हेल्प करते हैं? चेन लेंथ, सैचुरेशन एंड स्टीरॉल कॉन्सेंट्रेशन. ध्वनि,
Sorry, ma'am, could you repeat?
what I'm saying is, are these three parameters clear how they affect the membrane fluidity and how organisms would adapt with these three parameters?
Yes, ma'am, this is clear.
ठीक है, सत्यकाम बताओ
Yes, ma'am.
क्या नहीं क्लियर है. अब तो नाम लेके सबसे पूछेंगे. सब क्लियर है?
Yes, ma'am.
हाँ गोपिका, you want to say something?
Yeah, ma'am, could you repeat what sterols are like again?
What sterols are?
Yeah.
Huh? They
Yes,
are
ma'am.
okay, so they are these rigid structure. मैंने कहीं पे एक बार बनाया था, एक मिनट जरा रुकना. Yeah, this is an example of a cholesterol, but hopanoids or the other sterols present in prokaryotes also have these kind of a rigid structure. ये planar ऐसे 4-ringed structures होते हैं, which gives it the rigidity. And when it is so when the temperature is, let's say, low, the presence of these sterols, they break the interaction between the neighboring fatty acids, okay, thereby acting as a regulator. और एक जगह हम तुम्हें और बता रहे थे, कहाँ था वो, where is it, where I was showing that it's a dual role that it plays. हाँ, तो अगर तो ये सॉलिड ऑर्डर्ड को तो हमने बताया कि अगर मान लो लिक्विड टेम्परेचर बहुत लो है तो दे एक्ट एज बैरियर्स एंड इंट्रोड्यूस फ्लूइडिटी. लेकिन इफ इट्स अ वेरी लिक्विड स्ट्रक्चर, then it also acts as a support. These strong rigid things in the membrane would make it less disordered. It makes it more ordered. Okay, इसलिए हम इसको बाई डायरेक्शनल रेगुलेटर कहते हैं.
Okay.
ठीक है. Okay, Vishantan, any doubt? I think I've taken the names of everybody otherwise.
No, ma'am, it's clear.
It's clear. ठीक है. तो बाकी जो डिटेल्स हैं वो तो आपको नेक्स्ट आपके बायोकेमिस्ट्री क्लास में बताई जाएंगे. देन इफ देयर आर नो फर्दर डाउट्स देन आई वुड एंड द क्लास. ओके, ठीक है. एंड वी आर गोइंग टू मीट नाउ ऑन Tuesday. See you all, guys. Bye and a very happy weekend.
Thank you, ma'am.